question 10 of the gene predicting bioinformatics exercise. You used up to question 9 the ab initio gene prediction, which you were looking for cues of the DNA sequence that the computer will think that there's a gene here, there, or there. And now we want to see, okay, is there any evidence for me to believe that these are the sequence beyond the cues on the DNA sequence? When I say is there any evidence, what I'm looking for is, if I have this DNA sequence, does it match any protein sequence in a database? Because if it matches a protein sequence, once I do the 6-frame translations, maybe some of these will match, then that will say, oh, I have biological information that supports that there is a gene here or here on the genomic sequence. So on the FGenes-H that you used, the program predicted 2 different genes. And maybe you want to see, do we know, is there any sequence that matches either of those 2? And if so, can you find out the function? Can you find out what those sequences are? So now we're going to do what's called homology-based approach to find genes instead of an ab initio approach. And the tool that we're going to use is BLAST to do homology-based. And there are different ways you could go about this, and I will show you some of the ways. So we have this sequence for the Y chromosome. That's what we were using to look for predict genes. Let's see, do I still have? Yeah, I still have the Y chromosome. And you're like, how did you do that? I copied from. No, this was only the first 3,000 for another exercise. So I copied this whole sequence, and earlier I just looked on the NCBI website. So I looked on NCBI, I went here on the nucleotide, I looked for like Y chromosome, human. I found this record and I was like, okay, this is the one that I want my students to use. So I gathered this whole sequence all the way to the end. So all of that, and I copy that, and I'm going to paste that in BLAST. And there are different BLAST programs that I could run. So if my goal is to figure out if there are genes in this genomic sequence, then I have to be more selective about which database I'm going to search this against. So I could do this different ways. I could say, if this is DNA, then when it gets transcribed and translated and you have a protein, does the theoretical protein or the collection of theoretical proteins that are made out of this sequence, does any of those match a protein database? So in that case, I'm going to, instead of doing a BLAST N, don't do a BLAST P. So BLAST P you have, so it says using a protein query. I don't have a protein query. I could, if I wanted to do a bunch of work, take this sequence, throw it in a program to do the translations, and then I will take each of those 6 translations. So I'll do 6 BLAST searches using BLAST P. But I don't want to do that much work. I'll just make BLAST do the work for me. So here you search protein database using translated nucleotide query. Oh, that sounds good. So the query is the DNA sequence I have. That's the stuff I entered here. And so the program, okay, let me just hover over again. The program, I have a nucleotide query. The program is going to translate it for me and search the protein database. Okay, and I want to search all proteins. That sounds good. I don't want to narrow it down to anything. I just want to see, does it match any protein at all? And then I'll blast that. And while I was doing that, I want to open another window to do a different blast approach, which is what I was telling you earlier, that I'm going to be more selective about which sequences I'm going to search if I was doing a BLAST-N. So the BLAST-N, I have a nucleotide query and I'm looking for a nucleotide match, but I don't want to know what does this sequence match. I already know it's human Y chromosome. So that's not my question. My question is, does this match any mRNA? Because it has to match a DNA database, right? Because it searches a nucleotide database. So it has to match some sort of either DNA or RNA, so nucleotide. But to show that it's actually going to, this is transcribed in some way, I'm going to narrow this down to RNA sequences. Okay, I just want, or it could be EST. So this or this, they both are looking for, does any part of the genomic sequence match an RNA sequence from the database? So either one of these two. If I want to be overachiever, maybe I am an overachiever, and I'm going to search both, but I can't do both in one window. I'm going to do all of them, RNA and EST. So this is the days that we sequenced only the ends of the mRNA because we wanted some information, but the sequencing information was more expensive. That's part of why we only did the ends of mRNA. Okay, so let's see. My protein, my BLASTX is still running. I expected it to take a while because, you know, it translated to 6 frames, that's fast, but then it's taking each of those 6 frames and doing a search. So this window is doing 6 searches. This window is doing the nucleotide against nucleotide, only one search. And this one, I guess this one looked at the smallest collection. That's my guess, that's why it did faster. So then this. They're both nucleotide, but this just had fewer things to look through. So, okay, it is some mRNA sequence. So we could see where within that sequence that I entered, it matches an mRNA sequence, which gives me biological information that corroborates information about where the genes were predicted. Okay, corroborates if they match the same spot. But I do know that that sequence matches some mRNA sequence. Let's see how well it matches. Oh, matches really well. See expect 0. Remember, the lower this expected number, that's the e-value, the more meaningful that match, even if the identities are not 100%. My expected being so low, I'm going to believe that's a true match. That's a meaningful match. So let's see. And it looks like from the sequence that I entered, nucleotide 181 has a start of a gene. So that's plus plus. Okay, so F genes H didn't see that one. I think the first gene they predicted was in the 500, right? If I remember right. So this is not corroborating prediction. This is predicting something the other approach didn't predict. So for my PhD, when I was annotating strawberry, I was using both approaches and seeing where do they agree, where do they not agree. Let's see, then here's where some other gene happens, and I know that's a gene because it matches an mRNA sequence. And let's see this other one where I was looking. And sometimes it tells you what it is. So let's see, and I know it's from humans, and right now it's not telling me what it does. But what's more likely to tell me what it does is the protein one. Why there was a problem. All right, so I'm going to make some changes here and see if that's gonna fix that. It normally works, but I guess it didn't this time. But let me see. Let's look at the graphic summary. And then these are where, this is my query, and this is where there were matches. Interesting. It didn't predict the one that was like in the 500 and something here. And so let's see. Uncharacterized protein, just tell me what it does. So these are just saying it was matched some mRNA, but it's not telling me what the mRNA does. And oh, here, superoxide dismutase too. That's better in terms of it's telling me some role in humans. And had I not already gotten some other result earlier, I would just go with this. I had an educated guess of what that sequence was, and that's why I'm like, I'm not finding what I wanted. But if I didn't already know, I would say that's the role. I'm going to add this to the answer key. And that's how you would look for biological information to see where in this DNA sequence, in this genomic DNA sequence, there might be genes.